Hello everybody, I am Javier Cabello, a PhD student from Imperial College London, and today I'm going to tell you about my last work, Handhold-mediated strand displacement. If after hearing this mouthful you already want to stop the video, I don't blame you, but I can sum this up by telling that we have introduced for the first time templating in an autonomous DNA reaction. What this means and why this is relevant is because now we'll be able to catalyze the production of polymers with a specific sequence, just in the same way as RNA transcription or protein translation. Sounds good? Okay, let me explain this from the beginning. DNA, the molecule of life, encoding the wisdom of millions of years of evolution. But we don't really care about it. DNA nanotechnology forgets about all this gene business and focus mainly on the envelope. So we see every double helix as two wires that can bind by some kind of Velcro that we call Watson and Crick complementarity. During the last years, DNA nanotechnology has flourished mainly thanks to one reaction, Toehold-mediated strand displacement. This reaction is capable of spontaneously detaching a strand from a duplex with a target strand just by adding in the solution an invader strand. This reaction mechanism is mediated by a small sequence called Toehold. Toeholds are complementary to the invader and binds it while the displacement takes place. Eventually, the invader binds the target and the incumbent strand gets released. Toehold-mediated strand displacement has been used to implement a plethora of network elements in the same fashion as digital circuits, like logic gates or feedback loops. These elements have been already used to build chemical reaction networks with many different functions, such as drug delivery, sensoring, and well, many others. One of particular interest is the use of DNA to store information inside this sequence. Hitherto, many processes have been proposed to encode, read, and overwrite information inside DNA strands as if they were some kind of hard drives. But what about copying that information? Copying has never been autonomously implemented in a DNA network, and the main reason is that we still lack one vital function for copying, templating. But why is templating relevant to us? Okay, let's do a quick imagination exercise. Close your eyes and imagine that you need oxygen to live. Not very hard, right? To transport that oxygen, you need to produce a considerable amount of hemoglobin, a particular protein with a particular amino acid sequence. If you were going to build this protein, you will need to take the amino acids that form the hemoglobin and arrange them in a particular order. But there is a catch. Amino acids alone are incapable of binding in a particular order because the peptide bond between them is completely non-specific. So at equilibrium, not only our protein will get formed, but every single combination of amino acids of the same length will be equally probable. The situation gets particularly grim, especially for runners, when we consider the 20 standard amino acids at the same time. And the human hemoglobin, of course, is formed not only by two amino acids, but by hundreds. So how does biology evolve and conserve the sequence of a specific complex protein and express it at any time? With the help of templates, a molecule that can encode the same sequence information as the produced protein. So a strand of DNA can template a strand of RNA that can template the protein formation outside of equilibrium. This behavior is completely alien to most DNA reaction networks that just rely on equilibrium to push several strand displacement systems in a one-shot fashion. But what makes templating different? How does it really work? During a templated process, some sequence units, better known as monomers, this can be amino acids or nucleotides, produce specific recognition interactions with a template. These recognition interactions favor the addition of the complementary monomers by the ribosomal polymerase to a growing polymer. After the polymer gets produced, it must detach to be functional, breaking all recognition interactions with the template. This is also particularly useful to reuse the template. I mean, a one-use template wouldn't make too much sense. For that, you just assemble the protein in the first place. So, given the vital importance of templated copying in biological processes and for us to keep breathing, can we extract its basic principles to implement it in only a DNA reaction? Well, if we remove all the complex protein machinery, we can see that templating has two main requisites. First, the correct monomers have to react at a higher rate via specific recognition interactions. Second, once the whole reaction is completed, the produced polymer needs to detach. So our final objective will be to create a reaction that satisfies these two requisites, and after that, use it to autonomously copy DNA sequences, just from a solution containing complementary DNA strands that can detach after polymerizing. Toehold-mediated strand displacement looks already like a good candidate to implement templating. If you have two invaders equally complementary to a target binding domain, their reaction speed will be fully determined by Toehold complementarity. The Toehold acts in this case as the specific recognition element that discriminates which monomer binds. This is already great for our purposes. However, the Toehold is located adjacent to the binding domain between target and invader. This stable binding clamps the two strands together, preventing Toehold detachment after the strand displacement takes place. With this binding cooperativity in mind, a way to ensure the detachment of the strands would be to change the position of the Toehold. Different topologies, like the associative Toehold, introduce the recognition element in a completely different strand. In the same way as before, an invader strand with complementarity to this template strand will react quicker, and again, the template strand will stay firmly attached to the target and the invader. So we still need a topology capable of breaking recognition interactions after the displacement takes place. We still need something that makes the recognition interaction to break. Something like the incumbent strand. So what we propose is to introduce this recognition element in the incumbent strand. We call this element the handhold. The proposed mechanism of this handhold-mediated strand displacement has two steps. Now, and only for your eyes, I'm going to show you a simulation of the process using the latest Microsoft software. In the first place, an invader strand can reversibly bind to the handhold. Then, the invader will more probably start the displacement reaction. The rate of this migration can be accelerated by adding a small Toehold. The formation of the product destroys the bonds formed between target and incumbent strand, and if the handhold is small enough, the recognition interaction can break. To test that this works in the first place, we designed a series of experiments to demonstrate the two requisites of templating. First, that invaders with handhold complementarity can react at a faster rate, and second, that the handhold interactions can be broken after the displacement takes place. We first test handhold effect on the kinetics. To monitor the reaction, we designed a reporter duplex that can react with the output of the displacement reaction, producing a fluorescent signal. The result looks pretty much like this example. This particular case is a reaction with a small 2-nucleotide Toehold and a 7-nucleotide handhold, triggered by 4 different concentrations of invader strand. The obtained trajectories were fit to obtain the rates of migration and handhold detachment for each particular handhold and Toehold combination. So, does the handhold introduce any difference with a regular 2-nucleotide Toehold strand displacement? Well, believe it or not, there is a reaction going on in here. Let me put this in a scale. As you can see, an only Toehold-mediated reaction takes way longer to complete. Now we only need to define how much is way longer. We trigger several systems with handhold complementary invader strands. We test different combinations of handhold and Toehold lengths. When fitting this data, we observe the apparition of different kinetic regimes for the reaction depending on the Toehold and handhold lengths. Many of these have a second-order overall rate. However, there is a small subset, the one here, that has a first-order kinetic regime. This makes impossible the comparison between the rates. So we estimated the half-life for each reaction under the same conditions. So a short half-life corresponds to a quick reaction and a longer half-life to a slow one. From this graph, we can already obtain some information about the reaction. First, reaction speed is directly correlated with handhold length. Second, we confirm that the reaction can take place in the absence of a Toehold with rates similar to a 5-nucleotide Toehold-mediated strand displacement. And third is that for a handhold sequence, the handhold effect looks like saturating around 9 nucleotides. However, for handholds to be really effective, they should increase selectively only the rate of handhold complementary invaders. So when using invaders without handhold complementarity, the rate observed should be similar to the one that we had without any handhold present. So we repeated the experiments in the same conditions without handhold complementarity, and we obtained this beautiful inverted German flag, demonstrating that handhold presence do not alter the rate of Toehold-mediated strand displacement. If we calculate the difference between the half-lives of these two sets of experiments, we see that the contribution of handholds is especially relevant for Toeholds smaller than 3 nucleotides. In this highlighted region, we can see that we can obtain half-life differences of up to 4 orders of magnitude, which translated to reaction rates, it is up to 5 orders of magnitude. Let's realize about this. It is seconds against months. This is the time to make a coffee compared with the time to make a baby. So after demonstrating handhold-specific rate increase, now we need to check if handholds can be small enough to allow product detachment while still increase displacement rate. We proved this with two different experiments, a kinetic analysis and a native gel electrophoresis. The kinetic analysis consisted in adding a reporter after the displacement reaction finish. If the product detaches from the handhold, the reporter can snatch it and produce a fluorescence signal. We test this reaction, and as expected, the fluorescence production rate is inversely dependent of handhold length. A longer handhold results in a lower concentration of detached product at equilibrium, which of course translates to a slower rate. For very long handholds, like the 20 nucleotide handhold, the binding is so stable that no triggering of the reporter ever happens. We use the trajectories to fit an estimated equilibrium constant between attached and detached products in function of handhold length. And we confirm that for handholds longer than 8 nucleotides, the handhold binding becomes that stable that the 3-strand complexes become more prevalent. These results agree with the saturation length that we observed during the rate characterization. The presence of 3-stranded complexes are confirmed with a native polyacrylamide gel. Here are some controls where you can see the expected height at which the 3-strand complex, the target duplex, the product, and the free incumbent strand should appear for a 20 nucleotide handhold. As you can see, the 3-stranded complex is easy to identify because it hardly advances through the gel. So when we load the products of displacement reactions for different handhold lengths, what we observe is that for handholds longer than 8 nucleotides, the 3-strand complex start becoming more prevalent. So we can assume that for handholds smaller than 9 nucleotides, all the product is detached. Exactly the same result that we obtained from the kinetic analysis. Having demonstrated both templating requirements, the next logical step was to actually demonstrate the templating of a product out of equilibrium. For that task, we designed 3 different target duplexes, with 3 different 8 nucleotide handholds and the same 2 nucleotide Toehold. We made each one of these duplexes react simultaneously with 3 competing invader strands. The invaders were equally complementary to the target strand but had different handhold complementarity. To monitor the 3 competing reactions, we added 1 reporter for each possible product. Without any handhold, the solution at equilibrium should be equally populated by all the 3 products. However, we expect that by adding a handhold, the handhold complementary invader will clearly overcome the competing reactions, thus putting the cherry on top for the whole reaction characterization by demonstrating handhold templating out of the equilibrium. And as expected, the presence of handhold A templates consumption of invader A, the presence of handhold B templates the consumption of invader B, and, surprise, the presence of handhold C templates the consumption of invader C, thus demonstrating that we can specifically template out of equilibrium a product that contains an arbitrary handhold sequence. This demonstration with a DNA dynamic network is a small sample of what biology implements at a greater scale in complex biological systems. So that's all. As a take-home message, you should remember, first, that handholds are per se a new tool for fine-tuning strand displacement rates. Second, that this rate tuning is produced by a transient binding, so the product free energy is not modified. And third, that this transient binding clears the path of dynamic DNA nanotechnology to implement templating out of equilibrium in DNA networks. But our work do not finish here. As you remember, our objective is to implement an autonomous copying process in a DNA network. In a similar way to our last experiment, we will aim to produce a specific product among several competing reactions, but this time not from target duplexes, but from strands in solution. First, we will aim for the catalytic production of sequence dimers. And you know what is better than catalyzing dimers? Exactly, catalyzing polymers of any arbitrary length and sequence using longer templates. So I would also like to thank all the people that has made this work possible. Many thanks to Dr. Thomas Ouldridge and all the group of principals of biomolecular systems, especially to Dr. Bully Bai. Also thanks to Professor Guy-Bart-Stan and the group of control engineering in synthetic biology. Dr. Bai also happens to be part of this group, because one ration of group meetings is not enough for him. I would like to also thank our funders, the Royal Society, the Royal Academy of Engineering, and the European Research Council that has funded this project under the European Union's Horizon 2020 Research and Innovation Program. Finally, I would like to thank you for listening me till the end of this video. If you have any questions, suggestions, or simply fan mail, you can send it to my email address. See you next time!